Welcome to Roadmap to Residency, Evidence-Based Applications. So in this video, we will be talking about a very vast topic, and that is bacterial genetics and antibiotic resistance. So whenever we talk about bacterial genome, okay, we take the ideal E. coli as our sample organism. So the colored blocks above or below the line, okay, denote the orientation of genes. That is the direction of transcription, like this, okay. So the genes on opposite strands are close but not at exactly same location. So the E. coli genome is usually single circular genomes in bacteria. It's tightly packed with genes and they have short intergenic spacers. Okay, you can see very short spacers here. The genes sometimes are organized as operons. Now, operons are expressed as single polycistronic RNA. There are 600 operons in E. coli, which often encode proteins for related function of or pathway. Example, lac operon for lactose utilization. It also contains the DNA transposons, which are nearly 20 in the E. coli genome. So we take the ideal E. coli as our example. Now let's dive deeper. So the bacterial genomes also contain plasmids. Now plasmids are usually small and contain circular DNAs. They are independent in replication. They are often present in multiple copies and they carry genes with functions that are non-essential to bacteria. Example, antibiotic resistance, virulence factors, and enzymes to synthesize toxins. Now, not all bacteria have single circular chromosomes. For example, in case of Deinococcus radiodurans, it contains two circular chromosomes, and it also contains a megaplasmid and a plasmid too. In case of Borrelia, which causes Lyme disease, okay, it consists of small linear and circular plasmids. So it is very difficult to define a bacterial genome. So if a plasmid carries essential gene or genes, so we can consider it also as a part of the genome. Now, there is one concept of lateral or horizontal gene transfer, which is the movement of genes horizontally from one species to another, which can be mediated by either macrophages, plasmids, or transposons. Now, this is not generally very commonly tested in the examination, but this is the one basic concept that you might need to know. So how antibiotics work? So antibiotics work by inhibition of nucleic acid synthesis, for example, rifampicin, chloroquine, by inhibition of protein synthesis, for example, tetracyclines or chloramphenicol, by action on cell membrane like polyenes and polymyxin, or interference with enzyme system like sulfamethoxazole. It can act on the cell wall, for example, penicillin or vancomycin. Now, since this is how antibiotics work, now there may be some mechanism about antibiotic resistance too, which should be similar to these mechanisms. So how antibiotic resistance actually happens? So in a spontaneous DNA mutation, the bacterial DNA may mutate spontaneously. The drug-resistant tuberculosis arises this way. Now, in a form of microbial sex called transformation, one bacterium may take off DNA from another bacterium. The penicillin-resistant gonorrhea results from transformation. Remember these things. Remember the specific bacteria causing specific resistance due to some specific mechanisms. The resistance acquired from a small circle of DNA is called a plasmid that can flit from one type of bacterium to another. A single plasmid can provide a slew of different resistance. Now, this is the different mechanisms of the uh, antibiotic resistance. Now, there can be enzymatic drug inactivation like beta lactams. Okay. Now, these beta lactams can cause inactivation of the drug. There can be reduced permeability to the antibiotics. Now, there can be active efflux of the antibiotics. No, the antibiotics has entered the bacterial cell, but due to the active efflux, the antibiotics, they can just throw the antibiotic out. The antibiotic cannot act on the bacterial genome and all. There can be drug target modification. Okay. Now, this is how the antibiotic resistance spreads. Okay. So now, for example, the patient, okay, gets the antibiotic and develops resistance bacteria in his gut. Okay. Now, for example, here we have the family, here we have the animals, here we have the vegetable farm and all, we have the patients and all. Okay. Now, for example, George gets care at a hospital, nursing home, or other inpatient care facility. The resistant germs spread directly to other patients or indirectly on unclean hands of healthcare providers. Or the resistant bacteria is spread to other patients from surfaces within the healthcare community. And when the patients go home, then these patients can transfer the antibiotic resistance towards their family members. Also, for example, in case of drug-resistant bacteria, which can remain on the meat from the animals, and if the meat is consumed by the humans, the drug-resistant bacteria can spread to the humans. Now, so you can see that there can be various mechanisms behind which the antibiotic resistance antibiotic resistant bacteria can transfer from one organism to another organism, from one setting to another setting. Now, the discovery of antibiotics is one of the most important advancement in clinical medicine and public health. It has laid the foundation for a number of other advancements, including the ability to perform surgeries more safely. Many antibiotics are derived from other bacteria or fungi, example penicillin. This is because microbes use antibiotics to fight off other microbes. But the use of antibiotics and more broadly antimicrobials, which includes medications that target not only bacteria, but also viruses and fungi, has exploded in recent years. Now, antimicrobials have been used on an industrial scale, partially because of overprescription in humans, but more so because of routine use in farm animals. In fact, a good number of antimicrobials are excreted from humans and animals unchanged, and these get flushed into wastewater, which allows the pathogens to be perpetually exposed to antimicrobials. Now, in response to this enormous selective pressure, many pathogens have become highly resistant to antimicrobials. Now, when it comes to bacteria, generally speaking, there are four mechanisms for how they become resistant to antimicrobials. So the first mechanism is antibiotic inactivation or modification, which is where bacteria develop specific enzymes that destroy and inactivate antimicrobials. One example is beta-lactamase, which is a bacterial enzyme that destroys antimicrobials that contain a beta-lactam ring, example penicillin and cephalosporin. The second mechanism is the alteration of a target or binding site. An antibiotic that cannot bind anywhere is rendered useless. And you also have the protein efflux pump and also have the decreased permeability in the organisms that we have talked earlier. So the summary is that the mechanism of antibiotic resistance can be broadly classified into four categories. First, there is enzymatic modification of the antibiotic, including beta-lactamase, which cleaves the beta-lactam ring in penicillin and cephalosporin. This is the second mechanism of antibiotic resistance, is alteration of the target site. Third mechanism is use of efflux pump, and the fourth mechanism is the decreased entry in the organism. Thank you for watching. Keep studying.